All right, since you're all just kind of staring at me, I guess I'll get started. I hope that you guys have all had a great week. Remember that you have the Bureau questions for the practical immunology assignment due by 5 today. Many of you have already emailed those to me, so that's awesome. You can also see that there is an assignment for next week listed on the schedule. Basically for that assignment, I'm going to give you a list of potential papers, all of which have been discussed on a podcast episode. I'm going to have you read the paper, answer some questions about it, listen to the podcast episode about it, answer some more questions. I have it partially written and not completely finished, so I will finish it up today and have that for you by the end of the day today, and that will be due next week, so that way you'll have that available for the full week. So that will be the plan for that. Today we are going to talk about NK cells, as you can see here. This is sort of our last cell type that we need to talk about. You can see that we start talking about peripheral tolerance mechanisms next week, which really kind of segues into autoimmunity. I think of the first peripheral tolerance day as sort of our last basic immunology day, and then we go into clinical. For many people, they find that peripheral tolerance one clinical already, so we're right kind of at around that border point of between the sort of basic immunology and clinical immunology now. This one's still basic, but we're going to go into clinical stuff very soon. So I have kind of avoided NK cells quite a bit throughout the semester. And in thinking about NK cells and the way I want to kind of frame our thoughts about them, we need to go way back to the beginning of the semester and into thinking about innate versus adaptive immunity. And so this slide and the next slide are really ways that we are going to think about defining innate versus adaptive immunity. So based on what you've seen so far this semester, how would you define something as being part of an innate immune response? Yes, this slide and the next slide have it in, immunologist words, but I want to hear it sort of in your definition, in your words, what things define innate immunity and what things define adaptive immunity. Yep, David.
It is already there. They pre-exist and they don't change based on clinical history.
Okay, so the innate responses doesn't change between first and second infection. Would that summarize what you're saying?
Yeah.
Were you trying to get at another point there with that, or is this the only point you were trying to get at?
I was also trying to compare it to adaptive, which comes in later. And
Okay, so you also were saying here that this is early, right?
yes.
All right. And so with adaptive, we've got sort of improves with different exposure. And we've got late. So you got a couple things you're putting in there together. What other things might you put in terms of some of these definitions? Yeah, Roxy.
Broad versus specific.
Okay, broad. So broad response for innate.
Specific.
Okay, specific for adaptive. Okay, what else would we put in here? Oh, Miles.
Maybe that you can't survive without innate immunity, but with adaptive it's a little bit more fuzzy.
Okay, so yes, although I have a harder time knowing how I'm going to work that one into NK cells, so I'm not going to write that one down. But yes, that's a great point. All right. Other thoughts that you might have about this? Yeah, Jazz.
versus adaptive, which kind of has like almost like this curved slope.
Well, so I think that that's kind of what the not changing versus changing is really getting at with that. When we talk about broad versus specific that Roxy pointed out, can you give me any kind of mechanisms or processes that are particularly important for that distinction? Yeah, Emily.
Innate relies on pattern recognition receptors, and adaptive relies on antibodies and receptors.
Okay, and so we're going to say PRRs or kind of normal receptors, sort of standard types of receptors. Standard receptors. Okay. What kind of so Emily mentioned antibodies and receptors for adaptive. How do we get those? Yeah, Roxy.
Those activation of B and T cells.
Okay, so we're gonna have activation of B and T cells. I'm gonna come back to that in a second. Okay, what process do those activated, or actually before they're activated, what process do they do? What is it? VDJ recombination. So part of this whole specific thing is about VDJ recombination, right? Roxy used another really interesting term here. So Roxy, you just said the adaptive B and T lymphocytes. So how might that tie into this difference that we're showing here? Sometimes people, yeah, Jazz.
Kind of composes the innate versus adaptive. So adaptive involves B cells and T cells.
Okay, which are called
Lymphocytes.
lymphocytes, or they are part of the lymphoid lineage. Okay, what about innate? Yeah.
Neutrophils.
Uh-huh.
Macrophages and NK cells.
Well, we haven't talked about NK cells yet, but neutrophils, macrophages, dendritic cells. Is there a word that encompasses all of that? Yeah. Myeloid cells. So we've seen the myeloid cells doing a lot of work in the innate immune system. We've seen the lymphoid cells doing a lot of work in the adaptive immune system. When we've also talked about this all these changes between the different responses, just like we had this sort of important term of VDJ that I could put with specificity. There's a term I could put up here with this change over time. What term is that? Can you even think about what we talked about last time? Hopefully you remember it. Memory. So we have these responses that don't change between a first and second infection versus those that improve with exposures or show memory. We have early timing versus late timing. We have broad versus specific. And I'm even going to say that, and specific, of course, involves VDJ recombination, or a response. And we can also say that that is a response to a particular pathogen. And then versus sort of this more broad response to sort of broad groups of stimuli. And we say that the innate immune response involved myeloid cells, adaptive response involved the lymphoid cells. Yeah? Does this seem to go with what we've seen thus far this semester? All right. So we've also, you can see, of course, have this on this slide comparing and contrasting innate and adaptive immunity, or we can see sort of this slide that is talking about what's the definition of the adaptive immune response. And I guess Jazz had sort of mentioned it. I didn't really put it in here, but I'll mention sort of that clonal expansion for adaptive immunity in terms of the kinetics of the response. So we see that important clonal expansion in adaptive immunity. So we've got all these differences that we can see. So today we are talking about another cell type, which is called an NK cell. NK cells. And what our sort of question is going to be throughout this discussion is, where do NK cells fit? Are NK cells innate cells or adaptive cells? Is sort of going to be our question throughout all of this discussion of NK cells. And so that's why we have this nice sort of compare and contrast checkbox list that we just made about what's innate versus what's adaptive. So you can see the NK cells shown here, paralleled to T cells, and you will notice that those NK cells are cytotoxic. So, and we'll talk more about that cytotoxicity in some future slides. So we'll come back to the cytotoxicity in future slides. So one of the things that we can think about in terms of NK cells is where they originate from developmentally. So where do these NK cells come from? Because we've talked about the development of all of our other cell types and where they come from. And so if we actually look at this process of hematopoiesis, we see that NK cells come from the lymphoid precursor. Basically, the precursor has decided to either become a B cell or an NK/T cell, and then it might become an NK cell instead of a T cell. And so you can see here all the things that come from the myeloid precursor. Here are the things that come from the lymphoid precursor. You can also note that if we were to look at NK cells under a microscope, and you do a quick count, they actually get counted among the lymphocytes, because visually, under a microscope with histology, they look identical to lymphocytes, unless you're using markers for specific proteins that are unique on their surface. So based on all of this, would you think that NK cells are innate or adaptive? What does this say in terms of innate or adaptive? Yeah, Emily? Adaptive, because they're from the lymphoid precursor. So we got one vote for adaptive here. Okay. One thing that I actually forgot to tell you on the previous slide is I will just note that one thing that's important about NK cells is that they do have a unique protein on their surface that distinguishes them, called NK1.1. And they do not have a T cell receptor, or they also don't have CD3. So there actually are some T cells that also have NK1.1. They're weird T cells called NKT cells. They're the ones that are responding to lipid. Our NK cells don't have a T cell receptor, don't have CD3, but they do have this marker NK1.1. And so that's one of the ways that we distinguish them, is using this marker. So if we were doing our histology and had NK1.1, we might distinguish them. But if we're just doing kind of this straight right stain, then we would not be able to. Okay, so we got one vote for adaptive here. When we look at NK cells, one of the other really key things to think about is when NK cells tend to be active. They are particularly active against viral infection. And in the yellow shaded graph, you see virus replication. So here you can see virus starting to replicate early, going up to some kind of peak, and finally kind of going away. In green, you see sort of the classic innate immune cytokines and where they're being made. In red, you see the T cell response. In blue, you see when the NK cells are acting and when we see the most activity of those NK cells against the virus. One thing that you can note is that the NK cells are sort of around this time where we stop the increase of the virus and make it stay at this peak, while the adaptive cells are sort of happening right here where we're actually reducing from that peak. And so you can see that NK cells are providing a relatively early response to viral infection, happening, within the first couple of days. So if we go back to our compare and contrast, where does this put NK cells? Yeah, Roxy. So this looks like an innate response. Uh-oh, we got one vote for each. You can see, this is challenging. That's kind of the whole point. We can also think a little bit about the function of NK cells. If you had to guess from their name, NK cells, you already get a fair amount of understanding of their function, which is that NK cells, NK stands for natural killer, and NK cells kill stuff. And so you can see our NK cell here that is able to kill an injured cell or kill an infected cell. And so you can see that NK cell is able to sort of specifically kill this one cell that has a problem. In this case, it's infected. It could also be some kind of injured cell. So the function of an NK cell is to kill other cells. This slide is so close to being perfect, and it irks me a little bit every single time with the one issue. And so basically what I want you to see here is how NK cells do this killing, how they do their cytotoxicity. And the answer is that they secrete perforin and granzymes or interferon gamma. And the one thing that's not perfect about this figure is I wish that the interferon gamma and perforin and granzymes were going to this cell instead of out the other direction. It makes me very annoyed that they're going out the other direction when they should be going directly to this cell. But we have perforin and granzyme production, we have interferon gamma production as our functions of NK cells. And so that is in fact how NK cells mediate their cytotoxicity. So this doesn't really fall into our table perfectly, but we can sort of think about it for a second. How does this relate to other things that you have learned about this semester? Yeah, David.
T cells also secrete perforin and granzymes to kill viruses cells.
Yeah, this is the exact same mechanism that cytotoxic T cells use. So the details of how NK cells do their cytotoxicity are exactly the same as the details of CTLs. And so in that way, again, I don't have it here to really circle well on my compare contrast, but in that way, they sort of have some similarities to an adaptive immune cell. They're acting similarly to another cell we've talked about that is an adaptive immune cell. So you could maybe put that as a vote in the adaptive column. So then we kind of need, the thing that we haven't really gotten to yet is what's going on with sort of receptors and how you turn on an NK cell. And so that's where we're going to spend a bunch of time, is looking at how we turn on NK cells, what kind of receptors they have. One thing I will say is that NK cells can be activated by type 1 interferon. Type 1 interferon is interferon alpha or beta. So we have seen interferon gamma a couple times this semester with adaptive responses. Interferon gamma is a type 2 interferon, not a type 1. The type 1s are the ones we saw with the antiviral state and innate immunity early in the semester. So NK cells actually can receive that type 1 interferon, and that can actually trigger proliferation and activation of the NK cell. And so this is an innate immune cytokine, and this is a pretty broad response. It's sort of a broad response to anything that led to a type 1 interferon response. So that's somewhat more innate-like. But NK cells also have their own unique receptors. Because we remain very creative immunologists, we call them NK cell receptors. However, NK cell receptors are a bit different than some of the other types of receptors that we have seen before, because instead of there kind of being just like the one structure of the T cell receptor or the one structure of the B cell receptor, there are lots of different types of NK cell receptors. A T cell might have lots of copies of its one precious T cell receptor. A B cell is going to have one precious B cell receptor, though it might have multiple copies of it on its surface. The NK cell is going to have a whole bunch of receptors. It's not going to have just one NK cell receptor. It's going to have a whole bunch of different ones. So again, that's a very innate-like phenomenon as opposed to what we saw with the adaptive response. When we think about NK cell receptors, we can put them into a couple of different groups. And we can group them in a few different ways. So I can think of lots of ways that people group NK cell receptors. One of them is related to what kind of protein structure we see. So you can see that some of these are drawn as having a lot of immunoglobulin domains. Some of them are drawn as having some other types of domains. So we could put those in different families. You can see the ones with the immunoglobulin-like domains usually have KIR as part of their name. These ones that have another structure usually have some other type of naming. So that's one way we could divide up NK receptors. But the big way that we're going to think about dividing up NK receptors is into two broad groups. One type of these groups are called activating receptors, or NK activating receptors. The other type are called NK inhibitory receptors. You can notice from this figure that there are members of, it's not like the protein families, like all of one protein family is activating and all of the other one is inhibitory. You can see there are members of both protein families in both activating and inhibitory. So we're really just going to think about activating versus inhibitory as our two big groups. Shocking to no one, the activating receptors turn on the NK cell. They activate the NK cell, while the inhibitory receptors turn off the NK cell. The activating receptors all involve a signaling partner with a domain called an ITAM, which you have seen earlier this semester. It was in the CD79A and B, it was in CD3. It's involved in activating signals for different kinds of receptors. And so it's not a surprise that we see the ITAM with the NK activating receptors. In fact, the A in ITAM stands for activating. Alternatively, the NK receptors that are inhibitory NK receptors have a different domain in their cytosolic section, which is known as the ITIM. The ITIM also has a tyrosine that gets phosphorylated. That's what the T is for. But when that happens, we see inhibition of the cell. The cell gets turned off. In fact, the I in ITIM stands for inhibition. And so this is what we see in the inhibitory receptors. So as you can see, this is showing the NK receptors divided up by different protein families. And what you can see is that in many of these protein families, there are both activating and inhibitory members. Others are a little more specific, but we're just going to kind of think about our families as basically generally having both of these in them. And I'm going to tell you about a bunch of different specific examples as we go forward. The NK receptors are encoded by NK receptor genes. And so you can see some of the NK receptor genes listed here. And they are normal old genes. There is no VDJ recombination required for these genes. Instead, there's this sort of long section of the chromosome, of two chromosomes, chromosome 12 and 19, that have a bunch of these different receptors. I told you that MHC is the gene in humans that has the largest number of alleles. So there's a lot of variation between MHC from person to person. It turns out that after MHC, the next most is NK receptors. So there's also a lot of variation in NK receptor genes from person to person. We understand way less about what the heck is going on there. But it does seem like there's a lot of variability from person to person. And we have lots of these NK receptor genes in each individual. And each NK cell is sort of able to express a combination of different receptors. So you can see some NK cells here, and instead of being like a T cell that has one T cell receptor, the NK cells have many different NK receptors, some activating, some inhibitory, just a whole bunch of different things here. And so from what I've told you thus far about NK receptors and NK cells, where would you put NK cells in terms of innate versus adaptive responses now that we've learned about the receptors and the details of the receptors? Yep.
Adaptive.
Why would you put them in adaptive?
Because of the receptors.
Okay. What about the receptors? Does that mean innate doesn't have receptors?
It does have receptors, either P or R, but
Uh-huh.
this feels a little bit more specialized to me than what a PR is able to provide.
Okay. Yeah, what were you going to say, Roxy?
I feel like it's kind of like the innate where it's just like, because it either turns on or turns off between just like attack the cell or not.
So one thing to note is that there's no VDJ with these receptors. And there is VDJ, so there's a reason why I made sure we put it there, with the adaptive. So one thing is there's no VDJ, and I haven't told you yet what the receptors bind to, but as you will see, the receptors are generally not specific to an individual microbe. They're generally going to be responding to some kind of broad stimulus. And so in a lot of ways, these cells were these receptors look a lot like innate receptors, more so than adaptive receptors, because we can see multiple copies, multiple different receptors on one cell, which is not what we see in adaptive immunity where we have allelic exclusion. We don't have VDJ, and we have these sort of more broad receptors. And so we usually think of NK receptors as being much more like innate responses. And so I would say, at least in terms of what we're talking about right now with NK cells, again, we'll get into some other stuff as we go forward. Definitely when I started graduate school, for example, and we were like, NK cells, they are innate cells. They are part of the innate immune system. They're a little weird because they come from lymphoid precursor, but they're innate cells. And it was sort of cut and dry based on the early timing and the super broad response and the types of receptors. As I kind of implied in saying that, things happened while I was in graduate school. There was some additional discoveries that come back to this innate versus adaptive question. We're going to get to those later at the end today. So for now, we're going to kind of talk about this classic understanding of NK cells. I'll give you some of the more recent stuff a little bit more at the end. And as I mentioned in response to Jazz, the thing I haven't really told you yet is what's going on with the NK cell receptors. What exactly do the NK receptors bind? And our understanding of this really came from a specific set of experiments done by an investigator named Klaus Carr. And so we're going to talk about Klaus Carr's experiments. I'm going to tell you right now, I'm playing with fire today. The first time I was a TA, I was TAing for a person who actually knows Klaus Carr, and he drew this experiment wrong on the board. Because it is possible to draw it wrong. And so I usually have a crib sheet, and I don't have my crib sheet today. But we're going to do it right anyway. It's going to be okay. But what I say that because there are a couple things I'm going to emphasize as I'm talking about the experiment, and they're important to get in your notes, because if you don't get them in your notes and you look at your notes later, you're going to be real confused. So there are a couple things that we want to make sure that I'm going to emphasize that you want to make sure you really emphasize. So Klaus Carr had a couple of different strains of mice. One strain of mice had the H2D type of MHC. One strain of mice had the H2B type of MHC. And I don't exactly remember which one is in this case, but it doesn't actually matter for us right now. One of these mice had a tumor, okay? So we're going to say it's the H2B mouse has a tumor. So there's a tumor on the H2B mouse. Okay? He was able to take the tumor out of the mouse. And work with it in the lab. And eventually, the H2B mouse got over its tumor. So we're going to say so eventually, actually, no, hold on. So in this situation, the mouse does not get over its tumor. The mouse dies. The tumor lives. The thing to be aware of is when I'm talking about this experiment, sometimes the tumor dies and sometimes the mouse dies. And the first time I saw notes on this, somebody just wrote dies every time and didn't specify if it was the mouse or the tumor in different situations. And it was real confusing because they were like, what's going on? Why is everything just dies? And so you always want to be careful if I'm talking about the mouse or the tumor that dies here, okay? So he did this experiment. He also did an experiment where he took this tumor and he injected it into his H2D mouse. When that happened, the mouse lived and the tumor dies. This is due to CTL activity and the MHC mismatch. And sort of the idea that that would happen was already known. This wasn't surprising. Everybody knew this was going to happen. This is related to some of the things that go on in transplant immunology. And so we'll go into transplant immunology in more detail a little bit later. So right now, just believe me. MHC mismatch, CD8 T cells, get rid of it. Okay? So that kind of made sense. But then he did another experiment. And in his other experiment, he took the tumor and he made some mutations of the tumor. So he made a mutant version of the tumor. And he eventually learned that the mutant version of the tumor had no MHC. So he made a mutant version of the tumor in the lab. He didn't actually know at the beginning what the mutation was, but he eventually figured out it was a mutant MHC. As you can see in his title, it is H2 deficient. And he put this mutant tumor into an H2B mouse. So remember, the tumor used to be H2B. The tumor, if you put it in an H2B mouse normally, kills the mouse and the tumor lives. But if you make a version of the tumor that doesn't have MHC, now the mouse lives and the tumor dies. So somehow, now that this tumor is missing MHC, it's not that it has the wrong MHC, it's missing MHC completely, the mouse lives and the tumor dies. Yep, Miles.
Wait, so did they put the mutant tumor into an H2B mouse?
Yeah, an H2B mouse, yeah. And they did do the further experiment where they took the mutant tumor, they added back MHC, they put it in this mouse, and the mouse died and the tumor lived. So really, this was all about the fact that MHC was missing. And I'll just tell you, they did some experiments, and it wasn't CTLs. The tumor was not killed by CTLs. We didn't lose it by CTLs, which is what happened with the H2D. So what they found in this experiment is that there's some way that cells that are MHC deficient, that are MHC minus, are recognized. There seems to be some, at this point, unknown way that a cell that's missing MHC gets recognized and gets killed. And that's what they discovered in this experiment. So, the selective rejection of H2 deficient lymphoma variants suggests an alternative immune defense strategy. The fact that we killed stuff that didn't have MHC means there's some other type of immune response we didn't know about, is what he saw. And this sort of led to the formation of a hypothesis known as the missing self hypothesis, which you can see is the title here. The missing self hypothesis is basically that there is some kind of immune response that detects missing MHC, the missing self. So if MHC is missing, you have a way to detect that missing self. So it's not just that we're looking for self versus non-self, missing self. The loss of MHC can also be detected. And so if you had to guess based on this particular description, or based on this process, you will probably guess, and you would be correct, that the thing that's responding to missing self is NK cells, because this is the NK cell lecture. And you would be correct in that guess. But I want us to think a little bit for a second about what's going on with missing self before we come up with a thought about NK cells and exactly how NK cells are doing this process. So if we think back to MHC, remember that I told you about how self-peptides are always being presented. This is really key here. The idea is that we are presenting self-peptides to show that the cell is healthy. And if that process is messed up, if we ever stop presenting self-MHC, if the self becomes missing, that is an indication that there is a problem with the cell. Now, I'm saying MHC here, but in fact, when we talked about MHC earlier in the semester, we broke it down into MHC class 2 and MHC class 1. And when we think about NK cells and their ability to detect missing self, it's really only one of the two. NK cells actually don't care about both class 2 and class 1. They only care about one of them. Which one do you think they care about and why? Yeah, David.
MHC1, because that's the MHC that presents stuff that's inside the cell.
Okay, so MHC1 because it's the thing that presents inside the cell. Why else do you think that for this missing self hypothesis, class 1 would be really important? Yeah, Chrissy.
Because almost all cells should be presenting things.
Yeah, everybody has class 1. Does everybody have class 2? No, so if you killed all the cells that were missing class 2, you would kill like all the cells of the body. So NK cells and their ability to look for missing self are really looking for missing class 1. Sometimes this leads students to ask the question, what about red blood cells? I'm now very worried about my red blood cells getting killed by NK cells. We'll get there. But the idea is that really because everybody in the body should have class 1 and it should be being presented all the time, NK cells can look for MHC class 1. So this isn't really about looking for class 2, it's really about looking for class 1, which is an indication that the cell is healthy, there is presentation going on, and there's correct stuff going on in the cell. What might be happening if the cell stops presenting on class 1? Why might a cell stop presenting on class 1? You might have even seen a slide of it earlier in the semester. Yeah, Abigail.
We talked about certain viruses that prevent cells from filling immunology.
Yeah, lots of viruses try to hide from the immune system, and so they actually try to stop the cells they infect from presenting on class 1. So oftentimes, if a cell is not presenting on class 1, that cell is infected with a virus that's trying to hide. And so it seems like a good idea to kill that cell. The other reasons why a cell might stop presenting on class 1 are really because that cell is super stressed out or has something going wrong with it. Like if it's a cancer cell that's like trying to divide really fast and doing all sorts of wacky stuff, presenting on class 1 may be the least of its worries, and so it might stop presenting on class 1. So either there's a virus that's getting rid of the class 1 on the cell, or there's some kind of massive stressor on that cell that's making the cell lose class 1. And so the idea with NK cells is that NK cells are going to kill these cells that are missing class 1. So either our injured cells or our infected cells. And that leads a little bit to a question of how the heck do you recognize the lack of something? How do you have a receptor that recognizes there's no MHC, right? That seems weird in terms of thinking how you set that up with general receptors of how is there a receptor that's detecting no MHC? So what were you going to say, David?
I think they just kill everything that doesn't have MHC.
So, but how do they see that? They don't have eyes.
They're using their receptors, the
Okay, but how does a receptor show that MHC is missing? So it gets tricky. How does a receptor show that something's missing? Yeah, Paige. So the way that this actually works is that there is a receptor on the surface of NK cells that binds to MHC, and that receptor turns the cell off. It's a negative receptor. So basically, if there's no MHC, we can't turn the cell off. So this, is a little different than maybe what you normally think of as how you detect something. The idea here is that we have this inhibitory NK cell receptor. You can see in general our inhibitory NK cell receptors are binding class 1. And the idea is if the cell is making class 1 correctly, that's going to bind to the inhibitory receptor on the NK cell and turn the NK cell off. The cell is going to be like, go away, NK cell, I don't want you. And if there's no MHC, then it's not going to be able to turn the NK cell off. The NK cell is going to be able to kill. And so by and large, in all the examples I'm going to be telling you about, inhibitory NK receptors are binding to MHC class 1. You can see this process happening here. So we've got a bunch of different NK inhibitory receptors. If we actually look at those NK inhibitory receptors and where they bind to class 1, they bind to a section that's over here off to the side. It's not the same place where the T cell receptor binds, it's over here. And it actually doesn't care what the peptide is. It's really binding more to the structure of the MHC, while the T cell's over here looking at the peptide as well. So it's basically just, is there an MHC to be bound? So you don't necessarily need different ones for different MHCs or different peptides. You can just have one NK receptor, and any old MHC will inhibit and turn off that NK cell, thus allowing the NK cell to find a lack of MHC. With all of the NK receptors, there are a bunch of kind of interesting, unique, fun examples. And so I want to mention one other example right now. I'm just realizing there is another example that should go here, and I have it elsewhere. I'm going to put one other one here in a second too, because it does go in this same location. So there are a few things other than kind of standard MHC class 1 molecules that can bind to NK inhibitory receptors. One example of this is called, in humans, something called HLA-E. So if you see the word HLA-E, if you see the term HLA-E, what does it sound like? What do you think of when you hear HLA-E? What does HLA tell you? Yeah, Elizabeth. It's an MHC molecule from what species? From humans. So we got a human MHC. Okay, what does the E sort of tell you? Or what does the E remind you of? Yeah, Elizabeth. Class 1, it has one letter. So you hear HLA-E, you're like, oh, something about MHC class 1 in humans. And then you think, wait a minute, she said that MHC class 1 in humans is HLA-A, B, and C. She never told us E. We never heard of E before. E is not a normal MHC class 1 molecule. HLA-E does not present peptides from pathogens, but it's a protein that looks really similar to MHC class 1. It's MHC class 1 like, but it's not a normal MHC class 1 protein. It is in fact encoded in the same part of the chromosome as the MHC class 1 stuff. But HLA-E does have the ability to present peptides. It has the ability to present one kind of peptide, just one. And the peptide that HLA-E presents is a peptide that's shown here as a leader sequence for the other MHC proteins. All you need to worry about for this, if you think back to Bio 250, you have learned about leader sequence as actually called signal sequence. The thing that's important about it is it's involved in biosynthesis of proteins that go in the ER membrane. So this leader sequence is basically part of the biosynthesis of other MHC class 1 proteins. It's made during production of the other MHC class 1 proteins. HLA-E can present this leader sequence from other MHC class 1 molecules as basically a way to show that the cell is doing MHC biosynthesis correctly. So it's just sort of a measure of, here is my leader sequence from that protein biosynthesis. I was doing biosynthesis the way I was supposed to. And that's the only peptide HLA-E presents. HLA-E can bind to a particular NK receptor. As you can see, it's actually a protein combo of two receptors called NKG2A and CD94, and that inhibits an NK cell. So basically, a cell can say, I'm biosynthesizing MHC correctly, and that can inhibit an NK cell through this NKG2A receptor. I remember in graduate school when I was really working hard to memorize these, I decided that just thought that NKG2A was annoying because I would like A to stand for activating, but it's inhibitory. And so it just sort of irked me. That's how I remembered that it was the one that really irked me, because the A didn't stand for activating, it stood for inhibitory. There are other types of MHC class 1 like molecules that can also lead to inhibition of NK cells through some unique NK receptors. And this is the place where I realized, I'm like, why did I put those slides in the other spot when I could put them right here? Because they go right here. Like, what was I thinking? So I'm gonna like jump ahead for a second. Basically, what I'm telling you about in this jumping ahead is another weird molecule that is like MHC class 1, but isn't really MHC class 1. You're gonna see its name sounds like MHC class 1 again, just like HLA-E did. And again, the point of this is as a way to turn off NK cells, as a way to inhibit NK cells. So in this slide, you were seeing HLA-E. In this other slide, there is a protein called HLA-G. So you can see HLA-G up here at the top. HLA-G, again, is a protein. Again, is an MHC class 1 like protein. It's encoded in the MHC region of chromosome 6. You can hear from the name, it sounds like an MHC class 1. But HLA-G is largely expressed by the placenta. And so you can see this cell in purple, this placental cell in purple, making HLA-G, along with some other MHC class 1 molecules. And the placenta is going to make HLA-G so that any NK cells that come nearby are turned off. Basically, the placenta would like it so that mom's NK cells doesn't kill the fetus, thank you. And so, in fact, the placenta is full of HLA-G so that any of mom's NK cells that come around are going to be inhibited. Because we don't want any mistakes here with the NK cells being messed up. And so, in fact, if you look at what happens during pregnancy, here you can see kind of mom's uterine wall, and you can see the embryo. Implantation happens, and you actually get some cells from the trophoblast, from the embryo, that go into mom's blood vessels and invade so that they can get some of mom's blood. Those are those placental trophoblast cells, and they are all super covered with HLA-G. to make sure that none of mom's NK cells can hurt the baby or hurt the placenta or hurt anything else. And so this is a molecule that structurally looks just like MHC class 1. Its point is to trigger NK cell inhibitory receptors and turn off NK cells that come around. And the reason why it's so important in this case is because we have to keep a pregnancy. And so you can see, again, we do have some unique class 1-like molecules that are doing some pretty cool stuff. And so this is sort of the model of what we've talked about thus far in terms of our NK cells. So if we have a cell that has MHC on its surface, that MHC will bind to the NK inhibitory receptor that will inhibit the NK cell, and the NK cell will not kill. While we have a cell that doesn't have MHC on its surface, there's nothing for the inhibitory receptor to bind, and so in fact that NK cell is able to kill. You can also see I had to put some white boxes on this slide, because this slide I can't tell you the whole story yet, or haven't told you the whole story yet. And that's because we do also have this other group of NK cell receptors, which are known as the NK activating receptors. So we also have to think about what's going on with the NK activating receptors. This is a somewhat old slide of NK activating receptors, but it gets the point across that I want to get across. So you can see that in some cases, we can sort of describe what's going on with NK activating receptors pretty well. But in other cases, there's a lot of questions. There's a lot of question marks. And so I can't just say like broadly, like with NK inhibitory receptors, I can be like, binds MHC class 1. We got how this works. There's a lot we don't get about the NK activating receptors. So I can't give you like a broad way the NK activating receptors work. Instead, I'm going to give you some specific examples of NK activating receptors so you can see how those particular receptors work. But again, it's going to sort of be a little bit sort of variable or all over the place. And so I'm going to tell you two famous examples of NK activating receptors, what they bind and how they work. And just know that there are tons of NK activating receptors out there that one could talk about. One of our NK activating receptors is listed here as CD16. And CD16, as you can also see, is listed as an FC receptor, specifically an FC receptor that binds to gamma, IgG. So CD16 is an FC receptor for IgG. So one way that we can activate an NK cell is if there is IgG binding to some other type of cell, and that will lead to this cross-linking. So you can see this here. Here we've got a target cell. Notice it's got antigens on its surface. So notice this is a cell with the antigen on its surface. It's not free antigen. We've got a cell with the antigen on the surface because we're going to kill the cell. If it was free antigen, there would be nothing to be killed. We've got antibody binding. That was an IgG antibody. And we have this CD16, this FC receptor that binds to IgG on our NK cells. And that CD16 FC receptor turns on the NK cell and activates the NK cell to kill. It turns out you already learned this earlier in the semester. When you learned about the function of antibodies, I told you about antibody-dependent cellular cytotoxicity, or activation of NK cells. This is that. So our NK cell has this FC receptor on its surface. That FC receptor is an activating receptor. If there is antibody bound to the surface of some target cell, that NK cell will kill the target cell. very often when I think about this, I often think about target cells that are infected with viruses who are putting their viral proteins in the cell membrane because they need to make the membrane protein, like transmembrane proteins. So I think about this as a lot with enveloped viruses because there's going to be antigen on the cell surface a lot with those enveloped viruses. So there was a question. Miles, was it you? Or who had the question?
It was me.
Okay,
But
Emily.
It's going to be like, why would a virus be putting? Why would it be on the surface?
Yeah, so a lot of times the viral proteins are on the surface because for sort of biosynthesis reasons, they have to get in a membrane. And so it's just part of the synthesis of those viral proteins. So this ADCC through this NK cell receptor, this activating NK receptor is one kind of NK activation. The other NK activation that I'm going to tell you about is kind of similar to what you see over here on this top image, and it's going to involve this NK receptor called NKG2D. Okay, so you're going to see NKG2D on our next slide coming up. So we have this receptor, it's called NKG2D. NKG2D is an activating NK receptor. And NKG2D binds to a couple of different types of proteins. So this slide is showing you these proteins that are bound by NKG2D. One of that, so there's these proteins called the MYC proteins, either MYC-A or MYC-B, and there's also these proteins called the RAE1 proteins. So you can see them shown here. So basically the idea is if NKG2D on an NK cell encounters one of these proteins, MYC-A, MYC-B, or one of the RAE1 proteins, that activates the NK cell. The NK cell is going to kill. You also saw that exact same thing up here. So here's NKG2D, it's binding to its ligand, that's activating the NK cell. So that's what's going on here. And so what, of course, you might wonder is, well then, why the heck would why would these ever get made? What's the point of these? Why do these start getting made to turn on NK cells? And the answer is that these are all proteins that cells make when they are under a lot of stress. So when a cell is stressed, maybe because it's infected with a virus and the virus is taking all of its stuff and it can't do its normal translation because the virus is using all the ribosomes. Maybe because it is a tumor cell that's reproducing too fast. Maybe it's because there's some other problem with the cell. When that cell is under some kind of cell stress, it starts making these cell stress proteins, MYC-A, MYC-B, and RAE1. And so what you can see is that if we have a cell that is making these stress proteins, that will activate an NK cell, and the NK cell will kill that stressed cell. And you can see that happening here as well, where we've got sort of the NK cell binding to these stress receptors, and that's leading to activation. The NK cell is going to kill. And so this image that you see on the left is kind of like a very classic way of looking at this. You can be like, oh look, here's our healthy cell. It has MHC class 1. That inhibits the NK cell. It's also not stressed, so we don't have the activation, yay. Or here, we got no MHC and we do have activation, and so you can kind of see this comparison here. In reality, NK cells do math. And so in reality, NK cells are actually adding up how many inhibitory signals and how many activating signals they get. And if the inhibitories are more, they're inhibited. And if the activatings are more, they're activated. So it's not just like you only have activating or you only have inhibitory. It's really sort of this cell is looking at how many signals it gets of both activating and inhibitory, and whichever one it has more of wins in terms of whether that NK cell is getting turned on. So based on this, what is your guess about red blood cells? You all have red blood cells because you're all, here. So what's your guess about red blood cells in this process, in this situation? Yeah, David.
May not have any activatory or NK activatory proteins, so NK cells don't kill it, even though it might not have the inhibitory one.
Yeah, so there are no inhibitory receptors. Red blood cells don't have a way to inhibit NK cells, but they also have nothing on them that's going to activate the NK cell. So the NK cell can't actually get activated to kill the red blood cell. And so that's sort of how you don't need to worry about your red blood cells in this situation. And as this sort of implies, this is why it's so important that every NK cell has such a combination of receptors. So it can be basically adding up signals it's getting through all of those receptors. We actually do see that if you recall with MHC, I told you that with MHC, different MHC types are associated with good or bad prognosis with different infections. And then I also told you that NK receptors are also really variable from person to person. It also seems as though there are associations between different genotypes of NK receptor and outcomes in terms of different viral infections. And it seems as though you can have situations where your NK receptors are a good match for your MHC or a bad match for your MHC. So sometimes it's actually like if you have this combo, you're in good shape, and if you have this combo, you're in bad shape kind of thing. You can see some of these here. So like with HIV, if you have this one receptor called 3DS1, and you have this particular HLA type, then you're going to have decreased disease progression. And so it does seem like your NK receptor is also quite related to disease outcome. But again, there's a lot that we don't understand about how all of this completely works. So all of this that we've talked about, like I said, is kind of where we were through the majority of the time when I was in graduate school. And NK cells, I was pretty comfortable with NK cells as innate immune cells, with these receptors that were responding to kind of general situations, no VDJ, we're acting early, all this stuff. Here you can see that early activation happening, this response to type 1 interferon. And then right at the end of that time, some really interesting experiments came out. And so I'm going to tell you a little bit about these experiments, and we can think about exactly what they mean and sort of where this has brought us in the field. So one investigator did a lot of this work, but one thing that people looked at was they saw that if you looked at an NK cell that was responding to a particular virus, you could actually watch that NK cell undergo clonal expansion and contraction. And the resulting NK cell at the end of this was sort of, as it's called here, a long-lived experience NK cell. It has been changed by this experience. Some people will refer to it as an NK memory cell. And in fact, if you take that cell and you look at it in a dish, it has better function than the NK cell before. So it has gained function. It's like a memory cell in that way. If you put it into immunodeficient newborn mice, it protects them, while the earlier NK cell did not protect them. So again, this sort of said you can make memory NK cells. And so here you can see some other data on this. So again, investigators put some NK cells into a particular mouse that they could track, and infected that mouse with a virus. And you could see the number of NK cells, the percents of them in the blood, sort of increased and decreased through a perfect little clonal expansion kinetics contraction phase that we saw before. Looks just like an adaptive response. If you put, so here we're putting those NK cells into immunodeficient baby mice that don't have NK cells, and we're infecting them. If we just infect them with the virus, that we get what we see in blue, which is that they'll die. If we give them some naive NK cells or NK cells that don't have any experience, it helps a little. That's in red. And that's if we give them 10 to the fourth, so 10,000 naive NK cells. If we give them 100,000 naive NK cells, they do better, but still not great. But if we give them only 10 to the fourth, 10,000 of the memory NK cells, so that lower number of NK cells comparing with this red bar, or the red line in green, you can see those memory NK cells work better. So all these things sort of came together to give this idea that NK cells could lead to a memory response. And so you can sort of see this here. These NK cells seem to proliferate, make effector cell NK cells, contract, form long-lived memory cells, and have this ability to respond better the second time around to the microbe. And so why do you think this was kind of mind-blowing at the time? What does this sort of start to tell us and start to make a problem? Yeah, Emily.
It's a classic adaptive.
Yeah, this is like a super classic adaptive response. This is like part of our definition of adaptive response. And so now it's like, oh man, we were pretty good at putting these cells into the innate box. And now they are not participating. They are trying to be in the adaptive box, and this one's really hard to understand. This one, like, people really were like, now I don't know what to do. David, what were you gonna say?
How are they adapting when they don't have VDJ, so they don't have a
We're going there. And so, there sort of was this sort of issue in the field. And part of what this was sort of starting to tell us was that our perfect definitions of innate and adaptive aren't maybe exactly true. That there is sort of some gray area here of some things that kind of don't fit into these two perfect boxes. And so it's now pretty well accepted that, we used to think about there being this myeloid cell response, the NK response, and then an adaptive response that changed over time. And now we're like, no, the NK cell response also changes over time and like is its own thing. It's like its own in the thing that's in the middle. Like it's its own thing that's sort of has some characteristics of each. These things didn't evolve to be two totally separate things. We've already seen ways that they interact with each other. And in fact, we now see this NK response that sort of is a little bit in this bridge or in the middle part. Yep, Jazz.
Where do you find, I guess, the productivity in terms of kind of classifying it as one or the other?
Huh? Where do we find, sorry, I didn't.
Productivity in terms of what's the, I guess, general purpose of trying to.
I think some of it is to give us model, if you say, oh, it's adaptive with these similarities to CD8s, then then you can say, oh, I'm going to look at these things also in NK cells. So some of it's about knowing what to look at, when to look, things like that. Some of it is so you know what lecture to put it in or what chapter of the textbook to put it in. So there's sort of, how do you write about it in your papers, things like that. So what we have actually now found is that among the lymphoid cells, there are actually a whole bunch of cells that sit in between innate and adaptive cells. So basically, they're lymphoid cells, but they have a lot of properties of adaptive immune responses. And so you can see, I told you here at the beginning of the semester, lymphoid cells were cells of the adaptive immune system, but I had to put some little stars next to it. Because yeah, we get B cells, we get our normal kinds of T cells, but in fact, there's also this whole other list of types of immune cells that don't quite fit in, that are basically what are now known as you can see here, ILCs, or innate lymphoid cells. So we've got this whole group of cells called innate lymphoid cells that are cells that come from a lymphoid precursor but have a lot of innate functions. So it's not just the NK cell. We actually learned there's a whole big group of these kind of cells. I will say a little bit more about ILCs, and I will in fact address David's question of how you get memory without VDJ on Monday. So I will see you guys on Monday. Remember your assignment today by 5.